Hi everybody, it's Dr. A. So in this series, we're going to continue a little bit with Labware, but it's going to be all about microscopes. So we're going to look at microscopes and their use in the lab. And so we're going to start just with some basics. So a microscope magnifies small objects such as cells and tissues. And a compound light microscope, also known as a bright field microscope, is the most used microscope in clinical laboratories. It's composed of two magnifying lenses, the objective and the eyepiece or the ocular. So the total magnification of the microscope is the magnifying power of the eyepiece multiplied by the magnifying power of the objective in use. So the eyepiece is usually a 10x, so it magnifies it 10 times, and then the objectives is going to depend on which one you have on your microscope and which one you're using. So if you use the scanning objective, which is a 4x, then the total magnification is going to be the 4x times the 10x objective, so 4 times 10 is going to be 40x, so it is 40 times magnified. If you are at high power, which is a 40x objective lens, then it's going to be 10x for the ocular, 40x for the objective, 10 times 40 is 400, then the total magnification is 400. Then you also have resolving power for the microscope. So the resolving power is the limit of the usable magnification, and resolution indicates how small and how close individual objects can be and still be recognizable as two separate objects and not one. It doesn't just run together. We are used to maybe resolution when we talk about pixelation and high resolution or high definition TVs and computer screens and things like that and phones, and so it's the same idea. So something that is well resolved, you can tell the difference between two objects, barely or just resolved. They're kind of blending together, but you can see that there's a distinction. Not resolved, it all blends together as one. So the numerical aperture, or the NA, is an index or a measurement of the resolving power of the microscope. So it's the light gathering ability of the microscope's objective and is quantitatively expressed in terms of the NA. And so it's a measure of the number of highly diffracted image-forming light rays that can be captured by the objective. Of course, the more of the light rays it can capture, then the more resolving power it has and the more high-res or high-resolution images you will have. So the higher NA values will allow increasingly oblique rays to enter the objective front lens, producing a more highly resolved image or high resolution image. So the focal length is the distance from the object being viewed to the lens of the objective. It's also often called part of it is the working distance. That's from the bottom of the objective to the slide. But the magnification and the NA, the numerical aperture, are inscribed on each lens as a number. And so this is an example of a high resolution picture. So high definition, you can see a lot of details in that picture. And this would be a low resolution, low detail. So you can see very big pixels and stuff. So if you look on this picture here and you observe on the lenses of your microscope, you will see numbers etched there. So this is where you see the magnification that's etched in there, and the numerical aperture, and then the focal depth length there. The working distance is between the bottom of the objective and the stage there. So let's go over the parts of the microscope. So the framework of the microscope will include the base, the arm, and the stage. So the base is what you lay on the table. Sit on the table is usually where the light source is located also. The stage is where you put your slide or your specimen. So again, usually it's a slide of some sort. And then the arm is what holds the objectives and the ocular. So the illumination system components, so you have the light source, and it's usually in the base, and with the light source you have intensity controls, and there are different types of bulbs that can be used depending on the type of light that needs to be generated. And then so you have to know that when you turn the microscope on, you can turn light intensity up or down, so you can get it really bright or less bright. So it's, if you will, it has a dimmer switch, just like you can have on your lights at home. You can brighten and dim the light in the microscope. Then the condenser takes the light from the light source and focuses that beam of light from the light bulb onto the material that's under examination. So for us, it's going to generally be a slide. It focuses it up onto the slide. And then within the condenser, there's an aperture iris diaphragm. So it's right at the bottom of the condenser, under the lenses, but it's within the condenser body. And you can open and close this aperture diaphragm to let more or less light into the field of view. And so this is going to be important when we talk about the use of the microscope, because you can use that to adjust the light and to adjust contrast, depending if you're looking at a wet mount, like a urinalysis type specimen, versus a stain slide like a gram stain or a Wright stain. And there is also the possibility of having a field iris diaphragm in some microscopes, and so it's another iris diaphragm, and that one's over the light source, and so you can increase and decrease in the light, how much light comes through the light source with this field iris diaphragm. So there's pictures of some of the microscopes that we have in our lab. And so for our specific microscope, then here is the light source. You can see when you turn it on, this is where the light starts shining. And then the field diaphragm would be here if it had one. I don't think ours, I looked and I mean, it just doesn't have it, but I've seen microscopes and worked with microscopes where there is one. So there normally would be some kind of lever or a way to rotate this piece where it opens and closes and lets more or less light into from the light source into the condenser. So there's a condenser. We're underneath the stage here, and so there's a condenser, and then the aperture iris diaphragm is right here, and there's a little lever, and you can adjust it depending on if you're on 10x, 40x, 100x, depending. This is where you want to adjust it to. So if you look at it from the top of the stage, this is the condenser that you see here shining underneath your slide, if your slide is placed right here. And then here's a zoom in of the aperture iris diaphragm. You see the little lever. It's set on 40 right now, but if you were at 10x, you could go there and it would be less bright. And then if you are at 100x, you could go there, and that's actually completely opened with the maximum amount of light coming through. And this is, you can see it with the light on, so you can see how the light shines through the condenser, and it shines onto the objective through the slide, which there's no slide in this one. Oh, and then this is a video where you can see I'm adjusting, I'm playing with the lever. I'm going to play it again. I'm playing with the iris diaphragm lever in the condenser underneath, let me go back to it, underneath the stage here. So you can see very bright to dim to very bright. So there you go. All right, so the magnification system components, you have the ocular and the objectives. So the ocular is the eyepiece, is what you look through, right? And there is a lens in the ocular that magnifies the image that's formed by the objective. And we saw in the beginning of the lesson that generally the ocular magnifies 10 times. And then the objectives are, it can be achromatic or planochromatic. And then there's usually low power, high power, and oil immersion on most microscopes. Some of them will also have a scanning, with the 4x of scanning objective. And sometimes there's a couple versions of the oil immersions. It can be a 50x and 100x. Achromatic objective, the center of the field of view is sharp, but the edges are blurry. In a planochromatic, the field of view is in focus and flat throughout the field of view. The objectives are responsible for the primary image formation, so it's capturing the image from the slide, and they can help determine the quality of the image of the microscope. So, depending again on how good the objectives, numerical aperture, and all that are, and also obviously what magnification you're at. So in our own microscopes, these are the oculars, so the eyepieces that you will look through. They are adjustable, so you need to adjust them to the width of your eyes, because everybody, if you look, is a bit different. Some people have closer eyes, closer set eyes. Some people have wider set eyes, so you just adjust it to yourself. When you look through it, you should see just one field, just like a single circle, not two. And you can see here it is magnified 10 times, and so we have a magnification and numerical aperture here on it. And then these are some of the objectives. So the low power objectives, so this is scanning one right here, and then the 10x is the low power objective is a 10x. So if it says or instructions say look at it under low power, they mean for you to have the 10x objective down, you know, where that's the objective that you're using to capture the image. The piece that you can rotate to switch to different objectives is called a nose piece. And it is best to rotate the objectives by rotating the nose piece and not by grabbing the objectives. And then your high power objective is going to be your 40x. And you can see these are planochromatic lenses. 40x is a magnification, 0.65 is a numerical aperture, and it has some other numbers on there. This one's color-coded, and I believe this is pretty universal. At least all the microscopes I've seen seem to match this color scheme, where blue here is going to be 40x, and yellow is low power 10x. So this is high power. This is a dry lens. You're not supposed to use oil with this objective lens. And then we have oil immersion 100x planochromatic here, and this one is white, and so you have to use oil on the slide to use this objective. So this is the oil immersion objective. And then, so still part of the focusing system, you have interocular adjustment and left ocular adjustment, so you can adjust the width, how wide or close together the oculars are, but then you can also adjust on the left to adjust focus. You can rotate it and stuff. So then the body tube is a part of the microscope through which the light passes to the ocular, and the body tube length from the eyepiece to the objective lens is usually 160 millimeters. And the body tube is what actually conducts the image. And so what you have to know is that there is a mirror here. So the light goes straight, so comes here from the light source, goes through the condenser, through the specimen, straight through the objective, all the way in, and then it bends here. There's a mirror, and it goes through the body tube, and then that's what you see. And so the fact that you have this flip with the mirror is relevant in the use, because what happens then is, because what you're seeing is really a mirror image, when you move the slide to the right, your field of view moves to the left. And if you move it up, it moves down. So in the focusing system, you also have coarse and fine focus adjustment knobs. The coarse adjustment knobs will move the stage up and down quickly. They're the bigger knobs. I'm going to show you in a picture in a second what it looks like. You should always store your microscope with the stage all the way down, and so you would take the coarse adjustment knob and bring the stage all the way down. You would use the coarse adjustment knobs only with the low power objectives, so the scanning and low power, so 4x scanning and 10x low power. And then your fine focus adjustment knob moves the stage very slowly over just like one focal depth at a time, and you should only use it with a high power or the oil immersion objectives. So here are both sides of the microscope. You should have focus knobs on both sides of the microscope. So the one the closest to the body of the microscope, the bigger one of the two, is the coarse focus. So it moves that stage a lot. And then this one is the fine focus, and this one's the fine focus right there. So you can adjust from either side of the microscope depending on what you're doing, and if you need your hand. A lot of times, your hand might be adjusting the XY knobs, moving the slide around, and so then if your right hand is doing that, then your left hand can be adjusting the focus if needed. So let's talk about the passage of light in a microscope. So the lamp condenser, or the condenser, so the lens projects an image of the light source onto the plane of the condenser diaphragm. And then there's a substage lens system. So once the light has passed through the lamp condenser and the field diaphragm, then, so it's at the base right now, it passes through the condenser that's underneath the stage, and the condenser lens system, that's all that's beneath the stage, and that concentrates the light onto the specimen, which is on the stage, which is on the slide. Then the light, as it passes through the specimen, and then it hits the objective lens, which gathers the light that's passing through the specimen and projects the image onto the body tube, so it's into a mirror thing, a body tube, and that's the objective lens is the beginning of the magnification process. So depending on which lens you're using, it'll magnify it 4 times, 10 times, 40 times, 50 times, 100 times. And then the ocular lens is eyepiece, and it magnifies the image that was created by the objective lens a second time. Usually it's 10x, so 10 times more, and creates an image as if you'd close to the eye. So, and you just have to know that that is a mirror image, it's a flipped image. And so here it is again, the light is going from the path of the light from the light source through the condenser, focus onto the slide, grabbed by the objective, and put onto the body tube where this is reflected by our mirror, and then it goes through the body tube to the eyepiece there. So that's the path of light. So there we go. That wraps up our first part, and then follow me into the care and use of microscope for the next video.